for animal cloning. Group 2, Martin Gonzalez and Lucas McGarry. Animals and Society.
Martin Gonzalez, Birth of Animal Cloning. Cloning has been an unattainable dream. However, in 1996, cloning was achieved through animals, with the first clone being Dolly the sheep. Dolly's existence was a major landmark for science, as her creation shows the animals, such as mice and goats, can be cloned through cell science. This breakthrough in science also showcased potential, as animal cloning would allow people to have more animals for both business and medical purposes. Moreover, thanks to animal cloning, it quickly helped make laws and regulations so as to prevent using animal cloning research for human cloning through the Animal Scientific Procedure Act of 1986. Martin Gonzalez, Ethical Aspects of Animal Cloning. Thanks to animal cloning, many possibilities have been unlocked for the future of humanity, as it could solve many world problems. However, many ethical concerns rose over animal cloning and how abnormal it is. There are many arguments against animal cloning, however, it is often overblown, and there are many answers to these arguments. For instance, many people say that animal cloning hurts animals in the cloning process. However, the FDA assures the public by saying how many of these animal cloning risks are the same risks that assisted reproductive technologies and natural breeding face.
Lucas McGarry and Sociological Importance. Animal cloning is becoming a useful technique for producing farm animals and preserving endangered species. Cloning animals would allow for animals to have desirable traits. In 2008, the FDA concluded that meat and milk from clones of cattle, pigs, and goats are safe to eat. One possibility is you could clone your beloved family cat. There are many biotechnology companies in the United States and around the world that offer cat cloning services. Cloning animals can lead to advancement in medical technology and find effective treatments for diseases. The study of animal clones and clone cells could lead to greater understanding of the development of the embryo, of aging and age-related diseases. Current events related to the topic. A cloned black-footed ferret has given birth, becoming the first ever cloned animal of an endangered species in the United States to successfully produce offspring in what officials recently called a significant milestone in cloning technology. This marks the first time a cloned United States endangered species has produced offspring, showcasing a critical step forward in using cloning to enhance genetic diversity and conservation efforts. Black-footed ferrets are one of North America's most endangered mammals. According to the World Wildlife Fund, otherwise known as the WWF, habitat loss and disease are contributing factors to that. This species was once thought to be extinct. However, the species was rediscovered in Wyoming in 1981. Since then, conservationists have mounted concentrated efforts to give black-footed ferrets a fighting chance for survival. The goal is to increase the genetic diversity of the species to help them adapt to changing environmental conditions and disease outbreaks. This would have not been possible if it wasn't for cloning technology. clone pets. In South Korea, there was a dog named Snuppy that was hit by a car and tragically died. However, a group of South Korean researchers were able to clone him by using a single-cell code from the Afghan hound's ear. It was the first time a dog had been cloned in South Korea. This was a huge milestone for cloning technology. Now, this ability is extended to everyone if you have the means to pay for it. Companies like Biogen specialize in cloning cats and dogs. And if you look to the right, you can see the photo of the clone dog Snuppy. Current events. Today, researchers use animal cloning techniques to produce embryonic stem cells for many fields of research. Pluripotent, having the ability to develop into many different types of cells or tissues in the body. Stem cells are particularly interesting and improve our understanding of early embryogenesis, which is the process by which a fertilized egg develops into an embryo within the female reproductive tract, and stem cell biology, creating opportunities to develop stem cell therapies. Therapeutic cloning involves creating a cloned embryo for the sole purpose of producing embryonic stem cells with the same DNA as the donor cell. These are amazing cells, which have the unique ability to generate virtually any types of cells in an organism in order to grow healthy tissue in the laboratory that can be used to replace injured or diseased tissue. In addition, it may be possible to learn more about molecular causes of disease by studying embryonic stem cells and stem cell lines from cloned embryos derived from animals or humans with different diseases. Finally, differentiated tissues derived from embryonic stem cells are excellent tools to test new therapeutic drugs. Their discovery has greatly reduced the use of hurting animals in research. In addition, animal cloning research today is primarily focused on using cloning technology for specific applications in agriculture, conservation of endangered species, and even biomedical research, with a focus on improving the efficiency of the process and addressing potential health concerns associated with cloned animals, while the practice remains limited due to high costs and ethical considerations. Most notably, cloning is used to replicate animals with desirable traits like high milk production and livestock, or to even potentially revive endangered species through cloning techniques. But the overall success rate remains low, and further research is needed to address the challenges ahead. Cloning technology has improved since Dolly and will continue to improve through careful and ethical practices. Advancements in medical technology. Researchers have cloned many pigs with mutations in a gene that has recently been identified as a direct cause of Alzheimer's disease. This gives the researchers an opportunity to follow the early signs of the disease, as the pigs show changes in the same biomarkers that are used to make the diagnosis in humans. Researchers have studied the mutation of 3 genes that directly cause Alzheimer's disease. Through intense research over the past 20 years, it has now definitely been established that a mutation in a 4th gene, SORL1, can also directly cause the widespread dementia disorder. If this gene is defective, the person carrying the genetic defect will develop Alzheimer's. We know from human genetics that when the SORL1 gene is destroyed, we develop Alzheimer's. We have shown that if we destroy this gene in pigs, precisely the early changes occur in the animal's brain cells that we had dared to hope for. This makes it possible to find biomarkers that reflect the initial preclinical phases of the disease. Often, animal models are genetically engineered to carry disease-causing mutations in their genes, Creating these transgenic animals is a time-intensive process that requires trial and error and several generations of breeding. Cloning could help reduce the time needed to make a transgenic animal model, and the result would be a population of genetically identical animals for study. So this is a time-limited process, and by cloning, we can reduce that time. Positive effects of animal cloning in society. Positive effects of animal cloning in society include advancing medical research by creating genetically identical models for disease studies, improving livestock quality by cloning animals with desirable traits, preserving endangered species through cloning, developing new treatments and therapies by producing animals with specific genetic modifications to study disease, and potentially contributing to organ transplantation by creating genetically compatible organs for humans, all while allowing for a better understanding of genetics and development process. Cloning could be used to create better animal models of diseases, which could in turn lead to further progress in understanding by treating those diseases. It could even enhance biodiversity by ensuring the continuation of rare breeds and endangered species. As shown earlier in the slide with the black-footed ferret, by cloning animals, we can easily identify markers for disease in humans and those models to develop effective treatments. Reasons to be for cloning. Simply put, cloning equals the future. Cloning can help scientists study how disease works, how diseases progress, and develop new medicines. For example, scientists have cloned mice to study cancer and monkeys to develop new medicines for humans. Cloning can improve the genetic content of livestock species such as cows and pigs to produce more milk or meat. Cloning can produce animal models to study and treat human diseases. For example, sheep have been engineered to produce human insulin. Cloning could potentially bring back extinct species, like the woolly mammoth or giant panda. Gene cloning allows for making large number of copies of the desired gene in a matter of a few hours. Farm animals such as cows, sheep, and goats are being genetically engineered to produce drugs or proteins that are useful in medicine. Then they could insert a gene into the DNA of these cells that codes for a drug or a vaccine. If they take the nucleus from these cells and transfer it into a cow egg, it could develop a cow that makes drug in its milk. Since every cell of the cow would carry the drug gene, it could pass it on to its offspring. Even better, we could have drug-producing cows, and we could avoid the issue of genetic reshuffling that happens during sexual reproduction and simply clone our drug-producing cow. How to improve animal cloning. Educating the general public on topics related to biotechnology and how it benefits society. Using HDACI, histone deacetylase inhibitors, are a class of drug that target cancer cells by modifying gene expression. Compounds can increase the efficiency of somatic cell nuclear transfer, SCNT, cloning in mice, pigs, cattle, and rabbits. HDACI treatment improves cloning efficiency by enhancing gene expression and making chromatin more accessible to nuclear reprogramming factors. Removing epigenetic silencing marks. Researchers have found that removing epigenetic locks can increase can increase somatic cell nuclear transfer. Isolate desired DNA. By isolating the desired DNA and separating it, we can improve cloning results. By using databases, can help evaluate compositional equivalence and account for normal variations in a species. Some studies have used non-chemical and biological agents to improve the efficiency, and the technology has come a long way since Dolly was first cloned, and the tech is only getting better.
Martin Gonzalez, informed opinion and summary. In conclusion, animal cloning has a lot of potential for the future of society since its creation in 1996 with Dolly the sheep. With animal cloning, science has advanced as it provided the resources and data to learn about better medical practices that can be used to save people and animals from diseases and organ failure. Moreover, As time progresses and technology continues to improve, animal cloning has become better and much more widely available with companies such as Viagen. Viagen, they have been able to help people by cloning their pets and being able to reunite with them. Though animal cloning still has its flaws due to cloned animals not coming out perfect, it is a growing part of the industry and science that cannot be stopped.
Thank you for listening, and these are our sources.